Yes.
Okay, good afternoon to everyone. I want to welcome you in the next NGSeminar, and today's topic is proteomics, and our speaker is Pavel Sinitcyn, who will be talking about basics of proteomics, and also he will show us some of his achievements in this field. And Pavel is a postdoctoral researcher at the Max Planck Institute, and he worked for many years on application of many statistical and bioinformatic methods for proteomics and other high throughput assays. So he's an excellent speaker for this topic, and I want to thank him that he agreed to join our NGSeminars initiative. So Pavel, the floor is yours. You are welcome to show your presentation to us.
Thank you very much for presenting me. Yeah, today it was some small confusion, but rather which we resolved just yesterday. So initial title of the talk is Next Generation of Proteomics with MaxDIA, but while discussing yesterday, it's better would be to make some kind of introduction rather than myself, because I know why. So we decided to make just basically hitchhiking guide for proteomics, to proteomics, and just discuss, introduce terms which are common in this field. Lately discuss how can we apply this method in general, and maybe we can make a second part if it would, like some people will be interested. We're going to discuss actually application of MaxDIA. So there's a special type of proteomics called data-independent acquisition, but I have to say it's very complicated topic even for people in the field. So that's why I'm really thinking it's very important to make good introduction initially. Okay, let's go. Hitchhiking guide for proteomics. So this school is called NGS School or NGS Seminar. So it's a bit applying the fact that it's more about next generation sequencing. And as we know, next generation sequencing is more about genomics, transcriptomics field, like everything related to the new generation of like nucleotide sequencers. But I must say that proteomics is also a huge topic, and it has a huge amount of applications. And the development of proteomics, evolution of proteomics, I think, achieving the level of the current transcriptomics like state of arts. Not like single cell, let's say, we're soon gonna be there, but in general, proteomics is a way to go for many applications. Okay, let's discuss which one. So, but let's first of all compare these two omics, and we're gonna compare with like from different properties. And the properties like amplification. Do we have amplification in DNA and in the proteins? Of course, in DNA we do have, and it's very crucial step in sample preparation in many things, including single cell transcriptomics. But unfortunately, in proteomics we don't have. If you find, probably you'll get Nobel Prize. So complementarity rule. Do we have this rule in DNA? Of course, yes. It's a very famous discovery. So, but the problem is that in proteins we don't, and we don't have complementarity rule for amino acids. So this take into account these two factors, no amplification and no complementarity rule, like you can already consider the fact that we cannot use the same principles of sequencing, like a Sanger sequencing, for proteins, unfortunately. But there's you can wonder why not to use Oxford nanopore. Actually, many people successfully trying to use that, but there's another problem here. So as you know, Oxford nanopore even already have problems with 4 nucleotides. For that, you have to pass nucleotide sequence several times, or like use several tricks or some machine learning, which is very sensitive to small changes. So, but in the case of, it's already a problem to distinguish 4 different signals. But in the case of proteins, we have 20 different signals, and of course it's getting. On top of that, amino acids are very kind of varying in the sense of the chemical structure. And as a result, amino acids do have a lot of chemical modifications, and of course nucleotides as well, but it's not comparable to amount in the case of amino acids. And like also like if you think about this like as a as an information flow, as a genetical domain, a dogma of microbiology, we're passing from DNA, RNA, and proteins, and proteins do accumulate all degrees of freedom. It's basically integrative functions over the all previous steps. And the previous steps do have many degrees of freedom, like copy number variation, mutations, in case of RNA expression, splicing, secondary structure, allocation. But all of this is just kind of passing to next level. And it's of course, in the case of things like expression, of course, which we cannot express an expression which is hard to dig into using NGS methods. But there's something which is sure there's no way to study something like interaction, protein-protein interactions, cell localization, protein-cell, and post-translation modifications. But believe me or not, but all modern proteomics allow us to do that. And that's what I'm going to demonstrate today for my presentation. So one comment which I would like to give you right now. So if you have some questions, please feel free to write it whenever you watch, like either in YouTube or in another application. So just please feel free to write it. I think moderator will ask me, interrupt me, and ask me. So ask me during the presentation. It would be the best. Okay. So, but of course, like as in transcriptomics, we have like one very general problem, which is occurring from experiment to experiment. Imagine we have two biological samples, let's say cell lines, and one group of them is control, another group of experiment. And now I would like to find out. So I measured both of them. Let's say I measured with proteomics or some other methods related to proteins. And I would like to now find out which proteins do change expression, how strong. So this is basically the problem of differential expression. And let's ask ourselves which methods we do have in case of proteins. Of course, one of the most standard one is Western blot. So we are degrading sulfate bonds between proteins, within proteins. And next we are separating with electrophoresis. And the next using some staining, using antibodies or something else. We can really see do we have this protein in the sample and this how much, etc. So this is a very powerful method, but one of the biggest limitations of this method is we can study precisely 1 up to 5, let's say, proteins, not more. Okay, another method called 2D DIGE. It's actually different gel electrophoresis. That's where the name coming from. So this is a method which was extremely popular, let's say, 15 years ago. But this is, I think, still worth to mention because it's, I mean, in certain sense, it's a very beautiful method. So imagine that we have two samples, two Eppendorfs with, let's say, this Eppendorf is going to be control and this Eppendorf is going to be experiment. So we did our experiment. We did add some placebo to the control. And now what we're going to do, we're going to color all proteins in control with some red color, red dye, and in experiment we're gonna color all proteins with green dye. And next we're gonna just take all these proteins, mix them in one tube, and we're gonna proceed with 2 sequential electrophoresis. So first electrophoresis, let's call it like pH, acidic electrophoresis. We're gonna separate proteins through 1 T dimension in acidic to basic. And next we're gonna take this stripe and you're going to separate them with second dimension. Let's say the second dimension is going to be based on the size of the proteins. And why it's done, why it's 2 sequential electrophoresis, just to avoid the situation of the to really distribute proteins as much as we can. And of course, like we're gonna have like 3 scenarios at the end. We're gonna see like 3 type of dots. Either dots, yellow dots, it means that we had equal amount of red protein and green protein in both samples. Either we're gonna have purely green one, and this can mean different things. So it can mean that green protein overexpressed, or it can mean that green protein or protein in the control or protein in sample experiment was modified. So the result is change the properties and there's a two-tier comparison. So anyway, so whenever you see some dots which are not yellow, this is ringing it bell. So it's potentially differentially expressed proteins. So you're cutting out these proteins and sending this for mass spectrometry, and you're measuring that. So you don't need some fancy mass spectrometry in this case. You can use just MALDI, and this is very simple experiment design. You're just taking the protein, digesting it, and measuring with one shot of the proteomics. And yeah, just now the question is how many differentially expressed proteins you can check. Yeah, it's up to amount of time which you have for measurements. Let's say up to 100. Cool, to the DIGE method. So the next very important method in certain aspects and certain applications is method called CYTOF, or mass cytometry. TOF, it's from the name of the mass analyzer, time of flight mass analyzer, and cy from the cytometry. So what's happening in this method? Imagine that we do have very pure, we have very 50 tubes with in each tube we have separate metallic ion, let's say metallic crystals. It can be even not just some specific metal, it can be a specific isotope of specific metal, etc., etc., etc. But imagine that we somehow made 50 of them very pure, very nicely, like very small crystals. And now what's the idea that now we're gonna have also on top of having these 50 metals, we're gonna have 50 very specific antibodies against some specific proteins of interest. And this is a very good antibodies, very specific for certain proteins. And what we're gonna do, we're gonna take antibody by antibody, and gonna connect a special metal to first antibody, so another metal to the second antibody, a third metal to the third antibody, etc., etc., etc. So next, we're gonna mix all these antibodies with metal attached to them, and we're gonna incubate some cells inside of this mix. And as a result, if it was some protein X and we had some antibody against this protein X, so as a result, if like we see a lot of antibody attached to if we had a lot of protein X on the surface of the cell, then we're going to have a lot of antibody X on the surface of the cell. As a result, we're going to have a lot of metal on the surface of specific metal X on the surface of the cell. Okay, but how are we going to measure that? So for the measurement, we're using some very cruel methods. Let's say all these cells one by one dropped into the very hot plasma. So it's has a huge temperature inside. As a result, all organic chemistry is destroyed. But what is not destroyed, this metallics, so maybe metal is actually ionized, but what you can do next, you can basically just measure how many different metallic ions do you have the surface of the cell. So different amount of ions on the surface will report you how many different proteins you had on the surface of the cell. And this is basically done with very special time flight mass spectrometry, which we're going to discuss soon. But as you see, you're gonna have a matrix at the end with some amount of cells, some amount of rows, and honestly, maximum 50 elements in columns. So maximum 50 proteins in columns. So, but the coolness about this method is this method can measure huge amount of cells per second. It's basically something like 500, 1,000 cells per second. It's not instrument. It's really like some kind of mathematical weapon, which is taking cell by cell, pushing the cells into plasma, ionizing this metallic ions and measuring this, and doing this very quickly and very robust way. So, and how can we use that? As you may imagine, of course, this method really favors measurement of concentration of the membrane proteins, proteins on the surface. And that's actually true. Most of the methods doing that. And you can also measure a huge amount of single cells. So where it can be used, where the information about surface protein used like a lot, it's like That's, of course, immune system. So in case of immune system, just tell me which proteins do you have on the surface. I will tell you who you are. That's exactly why this method used a lot in pharmaceutical companies, which do have very good antibodies. Let's say we kind of selected some 50 membrane proteins, and we can measure these 50 proteins very reliably, very sensitively for some small changes over the millions of cells. And this is how it works really well. Because as I said, this method do have a membrane, like preference for membrane proteins, and it's extremely sensitive and it's so sensitive that this can work for single cell analysis. Okay, and this is one of the articles which just shows how powerful this method is. We just took these mice, we took different organs of these mice, and we measured the basically cell content, immune cell content of these organs. As we see in the blood, there's a lot of B cells. So of course, this was 50-dimensional space, and we converted this 50-dimensional space into 2-dimensional space. And now we see some clusters. We see clusters of B cells, T cells, and you can see how many different cells do you have in different organs. For example, in thymus, you obviously see there's a huge amount of T cells and almost no B cells, because we know that thymus is the kindergarten for all T cells in our body. And this actually works really nice, and why pharmaceutical companies do like it. The reason is because very quick and very cheap way of checking how a drug influencing on the immune system, or in general on body status, on physiology. That's why it's actually incredibly frequently used. Yeah. So if there's any questions, feel free to ask. Okay, so, but another question, like, okay, if optimize relative protein concentration. So we discussed a few free methods. Western blot, 2D DIGE, we discussed CYTOF, and of course today I'm going to discuss mostly about mass spectrometry, but let's discuss let's make comparative analysis. In the case of Western blot, we can analyze 1 to up to 5 proteins, let's say, but 5 is already something really a lot. Usually it's way less, 1 to 2. In the case of 2D DIGE, we can analyze up to how much time do we have, let's say 100 of proteins, but it's a matter of hundreds, let's say. In the case of CYTOF, we can analyze strictly 50, and the reason for this is because we cannot accumulate more than 50 pure isotopes and metals with heavy mass, which is not radioactive probably. And
there is a question to CYTOF actually. From Anna Maria. She wants to know if it only works for surface proteins or for others as well.
Yeah, that's actually a very good question. It's a question by which one of my colleagues actually participated in my course, and she worked a lot with CYTOF. And after the class, she came to me, and I said, of course, during the class, that it's a method which used for membrane proteins, strictly for membrane proteins. And after class, she came to me and said that's not fully correct. So this you can somehow make the cells permutable, so make holes inside of the cells, and you can incubate with antibodies. So this antibody will pass in of the cell. And I have to say that's not very common still, but people doing that. What people also do, people can extract nucleus, actually. They can extract nucleus and measure proteins in nucleus or on surface of nucleus. It's also very, well done and well established. But it's a very good question. Still, I think it would be fair to say there's still preference for membrane proteins. It's still the fact. Okay, so ability to distinguish small changes between all these four methods. In Western blot, we do know that Western blot works well in this very actually narrow dynamic range of the masses of the concentration, and as a result, I can say the ability is low. 2D DIGE as well. So of course, 2D DIGE has very high bias to the things which you can visualize, which you can see. You have very low concentration protein, and its change concentration, it's probably not going to be well visible. CYTOF is really extremely sensitive for small changes, and it's a fact. And mass spectrometry, the method which I'm going to discuss, it's actually highly sensitive, but I have to say it's just somewhere in between, you know. It's of course way higher compared to the Western blot, but it's about of site of close to this. But of course, from another point of view, you have to keep in mind it's in case of Western blot and in the case of CYTOF, we should have good antibodies, and that's something which you cannot avoid. And the CYTOF do has like this method does have preferences from membrane protein still. And the another methods which called another method, mass spectrometry, of course, one of the biggest advantage compared to all methods which just discussed with you, that's this method up to modern standards can really measure expression of up to 10,000 or even more. And this is actually very reliably done, especially reliably done with DIA method, which we're going to cover today as well, like in certain sense, like a bit. Okay, cool. If somebody will ask me to cover, give explanation what mass spectrometry is, to give definition and very quickly. So this is just a set of methods which allowed to take samples, and it's actually can be not just biological samples, it can be whatever, it can be actually even oil, and it's actually frequently used for the oil industry. It can be urine of the sportsman during the Olympic Games, etc., etc. So, and we're taking the sample and converting to the spectra. The spectra is just a function which looks like that. We have on one axis, or x-axis, we have mass to charge, and on y-axis we have intensity. And probably, if I tell you mass spectrometry, you already heard about the instrument. This instrument is used a lot, especially in airports. Yeah, for example, in airports, example, when I was flying recently from Marseille, there was kind of one officer came to me, he checked a piece of paper and put it in my luggage, and later like took this piece of paper and bring it to the instrument. This instrument was just a laser which was beaming to this piece of paper, and ionizing molecules inside. All these ionized molecules flying in the instrument, and this instrument is building such spectra, intensity, mass to charge. And what's the purpose of this instrument? This instrument will find out some intensity in, let's say, mass to charge 150. I say just out of my mind, 150, and 150, let's say it's trinitritol, very dangerous explosive. As a result, if this instrument will see some intensity in this mass to charge range, then that means that there's some danger going on. Of course, that's yeah. That's mass spectrometry used a lot in the airports, as I said, but also mass spectrometry used a lot in life science. And that's not something which is all time here. Let's say mass spectrometry in bioscience, life science is something which happened to be true from, let's say, 1980s, 90s, etc. So we're going to discuss actually why. Actually, yeah, we're going to discuss it even now. So that it was a Nobel Prize of 2002 for chemistry, and it's given for 3 scientists. But you can clusterize basically all these three scientists on 2 groups. So you may even see this based on the prize share. And one cluster is John Fan and Keiichi Tanaka. And these 2 scientists actually got the Nobel Prize for way development of soft desorption ionization methods for mass spectrometry analysis of biological macromolecules. Very complicated, but let's simple. So before this Nobel Prize, let's say before 1980s, it's like scientific community didn't know any method to take biological polymer and ionize it and transfer it from the liquid phase to the gas phase without actually destroying polymer structure. So let's say we have amino acids polymer, and we would like to transfer it to the gas phase and ionize it, but at the same time not destroy amino acids connections. This actually was a big deal. It was a big problem. It's not just about amino acids connections. It also can be nucleotide connections or sugar connections. And of course, that was no way to do that before. So you see, like something like this, it's like hot plasma. It was too hot, and it was destroying, fully destroying polymer structure, fully destroying all organic chemistry. This was not really feasible for manipulating with polymers, understanding the sequence the amino acids in the protein, etc., etc. So of course it was no way to go. And these two scientists, John Fan and Keiichi Tanaka, came to two different methods to actually do this, ionizing and transferring from one phase to another phase. And we're going to discuss it briefly soon. But it was also the third scientist, Kurt Wurtrich. Probably many of you do know him. And this person basically discovered NMR, so applied to the proteins especially. I mean, in general, NMR. Okay, cool. Okay, so Keiichi Tanaka and John Fan. So what's the Nobel Prize was given from them? So Keiichi Tanaka is famous for developing a method called MALDI. Probably many of you do know about this method. And this method looks like that. So we have sample on a metal plate. We discussed what can it be, but it's actually very broad application. In the old time, this would be just, proteins which we extracted from some sample and make some droplet and dry this droplet on the surface of metal. But in a general, you're taking laser and you're just beaming the laser with certain energy, and you are giving some additional energy to the sample, and this results into some proteins flying away from the sample. It can be proteins, either peptides, but that's, you see, we have these peptides and proteins are going to be charged. And this actually means that we solved the ionization. We transfer our sample from, let's say, solid phase or from liquid phase to the gas phase, and on top of that, we charged molecules. That's what's the purpose of the ionization in life science, let's say. Okay, but is it used a lot currently in the modern proteomics? The answer is not like, the answer is, it's a bit more complicated. So let's say MALDI today is used a lot in the image mass spectrometry, and let's see how it works. So it works approximately like that. Imagine you have a slice of the brain, and let's say mice brain, and what you can do, you can pixel by pixel go through this brain and ionize all molecules from certain millimeter by millimeter square. And by ionizing all these molecules, you will analyze all these molecules and find out how many different molecules you have. But the problem with this method, let's imagine that you had 10,000 different proteins in a certain region of the brain, and all these 10,000 proteins with different concentration will go on the gas phase, will be kept with a mass spectrometry instrument and will be analyzed sequentially. But the problem with this, that's 10,000 proteins will make a very complicated spectra. It's going to be very complicated spectra. Many proteins will misaligned against each other. We're going to have small mass differences, but not going to be able to distinguish them. As a result, this method, using very sophisticated data analysis software pipelines, and as a result, it can analyze, let's say, up to hundreds. And what's really cool about this method is we can see that we can see distribution of these hundreds of the sample. We can see that some protein, myelin basic protein isoform 4, see that this protein is located mostly in certain region of the brain, not over the whole brain, but in certain specific region. There's also something like protein like that. We see that it's some very specific region where it's located and nowhere else. And that's kind of what image MALDI can do today. That's actually, in this sense, it's a very powerful method for some. It can be brain, it can be any tissue, it can be cancer, it can be also plant. It works well. Okay, cool. The second part of the Nobel Prize for the soft ionization. So the first one was for MALDI, and the second one was for ElectroSpray ionization. So this part of the Nobel Prize was given to John Fan, and what is happening there? So imagine you have a small tube, let's call it actually even nozzle, and soon you will understand why, because we are bringing liquid through this nozzle, through this tube. We are pushing with certain pressure, let's say, but what's more important is this tube is heavily discharged. So we basically making big potential. We're making big potential difference between this tube, let's say the metallic tube, and between the instrument. So what's happening then? So all molecules which happen with liquid were deprived by electrons, and by constantly pushing, you're basically creating some kind of stream. You're making some clouds of droplets. Droplets are heavily positively charged, and what's cool happening here? That's this positively charged droplets. We actually, with time, by flying to the instrument, the positive charge. So, okay, in this positively charged droplets, there's a contents like a peptides or proteins and some liquid. So the media, let's say it's just water. And when this droplet is flying to the instrument, this water is just like evaporating from the droplet, and this droplet is shrinking. It's shrinking until a certain moment when the positive charge on the surface of this droplet starts really disrupting these droplets. And there's a really big discussion what's happening exactly in this moment. But the idea that this positive charge somehow transferred on the peptides or proteins, as a result, we are kind of the evaporation still going on, and this positively charged molecules are really just disrupting this small droplet with Coulomb power. And as a result, at a certain moment, you do have fewer proteins or fewer peptides which are positively charged. And as a result, you see, we solved this issue. We solved the issue of the transferring a sample from liquid phase, in this case, to the gas phase, and in the same time charging these peptides or proteins. But this ESI, electrospray ionization, has one huge advantage compared to the MALDI. One of the big advantage that's we still do have liquid phase here, and we can just really streamline some chromatography. Let's say HPLC chromatography just in the front of this nozzle. So, and this is actually what's happening in the lab. We do have in the front of each mass spectrometry instrument, we do have HPLC, and this HPLC, it's called high performance liquid chromatography. What it's doing is just basically separating proteins by some specific physical chemical property, let's say size, or it can be also hydrophobicity. So as a result, so the main point here, what we're trying to really avoid, we're trying to avoid a situation when we're going to have all 10,000 proteins going out of the tube simultaneously. How doing this? We are just separating all these proteins through the time and by some physical chemical property. So it's basically divide and conquer, if we speak in computer science terms. And by that, we can, in 1 second, we can analyze not whole 10,000 proteins, whole proteome, but we can analyze subset of this proteome. Let's say we can analyze 10 proteins. Let's say not 10 proteins, it's usually 200 something like this proteins. Cool. But okay, so but this we talked just about first step, ionization. So, and if you do have some questions, probably right now is the best time to ask. But while somebody may be typing, I will just summarize again. So 2 methods exist for ionizing, MALDI, which is using laser as an ionization source. It's basically adding energy to the sample, the evaporation with ionization happening simultaneously. MALDI is not used a lot currently in like very common proteomics when you would like to measure 10,000 proteins, but it's used a lot in the tissue-based image proteomics. We have also another method called ESI, which is a method which commonly used right now in all proteomics labs. And what this method is doing is just really making spray out of proteins, out of liquid and proteins, making small droplets. And these small droplets are evaporated, shrinking, and as a result is peptides or proteins at the end flying charged space. That's basically 2 methods for ionization, but of course ElectroSpray, huge advantage, disadvantage that we can couple with electrospray ionization, ionization with HPLC, with chromatography. That we don't need to analyze all 10,000 proteins at once, not all, like, let's say, millions of peptides at once, but we can really divide and conquer. We can separate them by time, and in each moment of time, we analyze not so many proteins. Yeah. So if some questions, so I don't see any questions. So then let's move on. Okay, so now we're coming to the mass analyzer, and there's a different way of doing this. But I also have to say this mass analyzer is very mature field, even like it's very old field. So even electron was discovered with very old mass analyzer by Thomson, but modern mass analyzer to the life science, to the analyzing, like, let's say, proteins, there's basically more or less two solutions which are commonly used in the field. First solution is time of flight, called, or even short, it's called very frequently TOF, time of flight. And another one is Orbitrap. Orbitrap is actually, I said this is a very old field, but Orbitrap was like discovered and created in 2000, let's say. And there's a breakthrough in our field. We're going to discuss first of all time previous interface, thanks to ionizer, whatever ionizer we used. We're going to have cloud of the charged peptides or proteins gas phase. And now we'd like to actually take this cloud and how many different species in this cloud do we have, and what is the mass of them, and how many do each mass we do have. A time of flight is basically, in general, consists of two parts: accelerator, so just electrostatic device, which is just pushing all positively charged molecules. Accelerator is also electrostatic, basically accelerator, putting in a side at 180 degrees, and not actually 180, but even 185, let's say. And what direction to the reflector? And in the reflector, your trajectory is bended, and at the end, you end up on detector. So detector is really very stupid. It's just measuring how many ions landed on me within a certain time period. And but actually, I have to say detector in time of flight extremely sensitive. Okay, cool. But what's really important to notice here that if you are a very light peptide or light protein, you pushed into the reflector, but because of you are very light, you don't have high impulse, you are really not flying deeply into the reflector. So your trajectory is very easy to change, and you are your trajectory quickly changed, and you end up in detector very early on. But if you're very heavy ion, thanks to accelerator, you're going to be pushed very deep into the reflector, and because of this, your trajectory is very long. You're going to end up in detector like longer after. And that's let's just check it out. Like it's like we can put some equation on that. So just like saying this accelerator gives some kinetic energy, and the reflector has some potential energy, which at the end equalizing kinetic energy of accelerator. And this pinpoint and this high point, these two energy going to be equalized, and that's why speed of the molecule here gonna be 0. Okay, cool. We can really equalize two things, and we can actually not so complicatedly. We can just find out that distance is constant. The electrostatic field is also constant. So as a result, we just Q, time, and mass is something which is vary a lot. So we can really just find out dependency of time from mass to charge. Why it's important? Because remember what detector is doing. Detector just bright plot. It's measuring how many ions, what's intensity basically, how many ions landed on me certain period of time. And using this equation dependency, which we know from like features of the instrument, we just can convert this graph, which is intensity from time, converts to the intensity to master charge. It's not really complicated. It's just following this equation. Of course, in real instrument, it's going to be way more complicated. But in simplific terms, you can think about time of flight exactly this way. And that's what we are getting. That's exactly what we are getting at the end. We're getting intensity over the master charge. This is what we call spectrum. Okay, but in the lab, in Mathematics department, you can find out, also in many labs actually, you can find a device like that. It's called the Bruker TeamSTOF, but the key word here is TOF. This instrument looks like that. So all samples, this is HPLC staining here. So all samples, first of all, going through the chromatography, all peptides or proteins. Next, we're coming here to the ElectroSpray. This ElectroSpray ionizing and ionizing molecules. And these molecules flying in the instrument like that. We're going first of all to accelerator, which is sitting here. And this accelerator is pushing all ions on top. And because why these ions not shooting, I mean, not landing on the roof, and the reason on the sail, the reason because we do have reflector here on top of the instrument. And this reflector is changing trajectory of the ions. These ions are going down, back, and going on the detector. And this detector is just detecting how many ions landed on me within the time. That's very simple, but there's actually some tricks related to this. So one thing, on one conference, on one workshops, I have been asked like some tricky question. Imagine that we calibrated this instrument. Calibration, what does it mean? That if we give this to this sample, to this experiment, if you give this device a sample consisting of different molecules, let's say mass of 100, 200, and 300 in certain concentration ratios, we kind of expect that this instrument will build us a spectrum, which is going to have masses 100, 200, and 300 with expected intensities. But nobody actually guarantees us that if something goes wrong with instrument, instrument can be wrong in different ways, including actually mass errors. And the truth is, let's say, imagine that we took like temperature 15 in the lab, 15 Celsius, and we calibrated with this very cool sample. We calibrated our instrument. Now our instrument really shows masses 100, 200, 300. Cool. But what we're going to do next? Imagine that's, I don't know, the heating system broke this lab. And now we, instead of 15 Celsius, we're going to have 25 Celsius, like on the street. And the question is, what's going to happen with all these molecules from the perspective of the instrument? Are we going to still see molecules with 100, 200, 300, or it's going to be higher masses, let's say 101, 201, 301, or it's going to be low masses? So it's actually an open question. And when teacher asked this question, actually I was mistakenly saying that masses will decrease. And the reason because by increasing energy, increasing temperature, you will increase amount of chaotic movement. As a result, like most of the ions will reach detector faster. But it's actually mistakenly thinking about, like it's not correct answer for this question. So the correct answer would be that all ions will get heavier. It's exactly what means that ion which was 100 will get like mass 101, 200 and get 201, etc., etc. And why it's happening? That's the most interesting question. So the reason for that, because as you see, this whole device is made out of metal. Also the tube, which is maintaining the very low pressure side, is also metallic tube. The truth is that when you're increasing the temperature, this metallic tube starts like expanding because the metal starts expanding thanks to the temperature. As a result, this tube is getting a bit longer. As a result, this distance which a molecule are flying is getting a bit higher. As a result, like all molecules will go and travel longer. As a result, from perspective of the detector, all molecules start getting a bit more heavier. This is actually the correct answer. Okay, cool. The second mass analyzer, which, okay, if there's some questions about TOF, I think that's now it's correct time to ask. Okay, while you kind of maybe typing questions, so I also describe you make some small introduction to the Orbitrap. So the truth is that time flight was extremely popular, let's say, until 2005. In 2005, the first device with Orbitrap on board was good, and that's something which changed the field completely. So many people start using Orbitrap, and the main reason why Orbitrap was so successful because resolution power Orbitrap is incredibly higher, way higher compared to the light instrument. But now I think we're very in a good position when we do have a competition of two companies who are actually providing different mass analyzers. So one of the company, let's say Bruker, which is one of the main company, and also Thermo Fisher. So Thermo Fisher doing a lot on the level of Orbitrap. Right now, there's a, let's say, big internal competition happening between two approaches. And yeah, and who getting most of advantage, of course, users. Okay, the principle of Orbitrap is way more complicated, let's say, and I think for the sake of time today, I'm not going to cover this so much, but let me describe the power of this method. And the power of this method can be visualized with like, yeah, that's how it looks in the lab. So we have like basically some box, which is the most expensive part of the lab, and this box is where the Orbitrap is sitting. This is a very small device, mass analyzer. But what's cool about this device? So imagine that we prepare samples which consist of just specific peptide, this specific peptide, and this peptide has a charge 3+. Let's say approximately mass of this peptide is something like 1,800 something, few cents. So if you divide by 3, this should be some value like this. It's going to be something 600.66666. Okay, cool. But that we also measure this peptide with, let's say, with different resolution settings. We can measure with very low resolution. It's some specific value, but let's just make sense of this. So 1,000 is very low resolution. 100 it's something 100,000 is something which is used a lot, and general resolution which is used now, and 1 million resolution is something very extreme. Some lab can do that, but it's very, you have to keep in mind that's very extreme. But the truth is that Orbitrap is that device which allows to make that, and for general public. It was another mass analysis which allows you to do exactly even higher resolution, but Orbitrap, which was something which was on the market. But okay, cool. First question which I may ask, see such peptide with charge 3+, and as a result, we do understand this master charge should be exactly approximately 1,800 divided by 3 is going to be something 600 around this, and we do see this. But the problem is, which I also would like to ask you, so why do we see not just one peak? Why do we see several peaks instead of just one peak? So we do have one peptide, charge of 3+, but instead of seeing just one peak with certain mass, we do see several peaks. And the main reason for this, it's natural isotopes. Because that's like if you look at the carbon, for example, like if you take 100% carbons on our planet, let's say approximately 90% of the carbon is carbon C12. No, it's not because of fragments. Actually, we are not coming to fragments yet. We are on a level of the peptide itself. So it's like, really, believe me, all these peaks is the same sequence of the peptide, but somehow we see not one peak, but we see several peaks. And I will describe you why. So imagine that on our planet we have carbon, and it's 100% of carbon. And the truth is that 99% of this carbon is actually carbon-12. But we also have some small portion, let's say 1%, actually carbon-13. So, and the reason is just like it's carbon which has additional neutron. It's basically 12 plus one additional neutron. The truth is that when you are looking at the peptide is huge molecules. And we do have a lot of the carbons, we do have a lot of hydrogens, we have a lot of oxygens, etc. As a result, the probability that you're going to have at least one isotope is actually not very low. It's actually, in this case, it's going to be at least 1%. It's also, you may by chance have two additional neutrons landing somewhere, you can have 3 additional neutrons, 4 and 5, etc., etc. So this is actually what we see. This is especially true, for example, for sulfur. In case of sulfur, one of the isotope is very abundant. It's actually something like 5%. Result, we can really see on the high resolution that's each peptide with sulfur in the content really splitting on two. It's actually very useful. But okay, we see that's in the resolution of low resolution 1000, which is not used anymore. Let's say we cannot distinguish different isotopes. Actually, sorry, maybe that's very important to describe you. So what's the distance between different peaks? If you look closely, let's say this peak is happening on 601. We have 1, 2, 3, and third peak is happening 602. By that, you can conclude this distance between peaks is 0.33333. And it totally makes sense because the charge of this peptide is 3 plus. So adding plus 1 neutron, you're dividing by 3. As a result, you're getting exactly this mass difference as 0.33. And as a result, you may actually make a reverse task by seeing the spectrum, predict what is actually mass of the peptide, not charged mass of peptide. And it's very easy to do. You're just taking one isotopic peak without any additional neutrons. You calculate the distance between two neighboring peaks. And if the distance is, let's say, 0.5, then it's obviously that's the charge of this peptide is 2. It's the distance, let's say, 0.25. It's obviously the charge of this peptide is 4. What you can do next, you just can take this mass, very accurately estimated mass, and multiply by charge. By that, you're going to get exactly mass of the peptide which was before. It actually works really nice, and it's actually used even a lot in the case of identification. Probably discuss later. Okay, cool. What we can also look, so you see these resolutions. 1000 is very low, 100 is higher, 1 million is something extreme, but also we cannot see any difference between 100 and 1000. I mean, 100,000 and 1 million, yeah? Almost no differences. But the truth is that let's zoom in for this peak. As you see, this peak has additional two neutrons. So this is 0 additional neutrons plus 1 isotope, so plus 1 neutron plus 2 neutrons. The question is, where these neutrons happen to be? And the cool thing that's we can zoom in and we can find out that we can see all possible combinations of the isotopes. We see that there's one peak which contains 2C13, another peak contains C13 nitrogen, there's one peak with oxygen 18, and etc., etc. So it's actually very cool, and especially what's cool about that is look at what's happening on the level of row, on level of axis. We can see that's actually this difference which you can distinguish. It's 1,000th of the Dalton. It is actually of Thomson, of course. Say, but this is actually dramatically huge, dramatically small difference, but we still can distinguish that. Just think about this, like these peptides do have the same amount of neutrons, they do have the same amount of protons, and a kind of naive brain, you may think about that the mass should be exactly the same. But it's not true, because the energy of system, when you add like neutron in the background of carbon, or you add neutron in the background of nitrogen, the energy of the system going to be different, as all the mass is going to be different. That's actually what we see here with very extreme mass spectrometry. But honestly, this 1 million mass spectrometry is something which we kind of now can easily reach thanks to the like modern Orbitrap mass analyzer. The next question which you can ask: how can we use that? Yeah, how can we use it for some applications? So there's some people who are actually doing the multiplexing based on this fact, but it's something like really, let's say, a bit minor. But yeah, you can also do some I don't know, carbon dating of the samples. Maybe it's can be useful in paleoproteomics. Yeah. Okay, cool. Maybe you noticed I several times I was correcting myself. I was saying we can analyze peptides or we can analyze proteins. And the truth is that this correction is highlighting the fact that we do have in our field two types of proteomics. We call it shotgun proteomics and top-down proteomics. And the difference is exactly the same difference. It's actually the dictionary terms are coming not from proteomics, they're coming from genomics. And in the case of shotgun genomics, as you know, we are not analyzing whole chromosome from beginning until the end. But before that, we are fragmenting because we don't have this technical capabilities to do so. But what we are doing, we're actually taking this chromosome, digesting it somehow randomly on a pieces of 1,000, let's say, nucleotides. And next, we're analyzing each piece independently from each other. So it's exactly the same principle used in the case of proteomics and shotgun proteomics. We're taking proteins and using, in this case, proteases, let's say trypsin, we are digesting these proteins on the peptides. And next, we're just taking these peptides and analyzing all together. So result, instead of, let's say, 10,000 of proteins, we're analyzing now millions of peptides. And yeah, so that's what shotgun proteomics is. But in case of top-down proteomics, we are actually taking the whole proteins and dropping these proteins inside of the instrument. And the whole fragmentation is happening inside of the instruments. It's actually not very right now important, but that's basically means that we are taking native proteins and analyzing with top-down proteomics. So what's the advantage of one compared to another? Top-down proteomics is a very cool thing, but up to the modern standards, we can analyze up to, let's say, very low complexity mixture. We can analyze a mixture of the 5 proteins, like 5 proteins. That's what's actually we can do, what's actually many pharmaceutical companies do, but not more, unfortunately. And you have to also keep in mind that top-down proteomics have very strict requirements for the instrumentation. But what's the method of choice for exactly the task of analyzing 10,000 proteins? It's the shotgun. And the reason, because it's way easier to deal with peptides. There's a less strict requirements for the mass-spectrometry instrument for the analyzing peptides. Because we're shot, chromatography is also very simple. You don't need such a huge vacuum for the inside of the instrument. And yeah, that's why shotgun proteomics is very, very popular. So since this moment, I'm probably gonna talk uniquely about shotgun proteomics. But there's certain specific topics where actually top-down can be useful as well. Topics like, let's say, occurrence of different modifications on the proteins or quality control on the production line. Let's say your pharmaceutical companies and producing antibodies. And yeah, that's where top-down proteomics is extremely useful. Okay, up to now, what we understand that we can take some human tissue, we can take some samples from human, and we can take proteins out, and we can digest, let's say, with trypsin, all these proteins on the peptides. And because we don't want to analyze all these peptides at once, we are separating these peptides using some chromatography, let's say HPLC. And next, all these ions coming to the ElectroSpray. ElectroSpray is transferring sample peptides from the liquid phase to the gas phase and simultaneously is charging them. And the next portion of the charged molecules, in our case, charged peptides, flying to the instrument, and we are kind of measuring the set of the peptides, and we are measuring basically spectrum. Let's say we are measuring spectrum. On measuring spectrum, we're spending 20 milliseconds of each second. So basically, it's pretty enough time. So we are collecting these peptides and measuring all together at once. So cool. And as a result, doing this every second, spending first 20 milliseconds of each second, we can really see that some peptides start disappearing from the column. So it's like we were in really high peak at a certain moment. Now we're going down. We also see that some peptides are totally disappearing, but we see also some, for example, this orange one, it's just appearing from the column. It's just like really start leaving the column. This is just here. Okay, cool. So as a result, we see such waves, you know, of appearance, disappearance of the different peptides. We also see isotopic structure. And thanks to the seeing isotopic structure, we can see the distance between peaks, neighboring peaks. By measuring distance between neighboring peaks, we can measure the we can understand what is the actual mass of the peptide. And by knowing mass of the peptide, can we actually get to know what's the sequence of the peptide? So I think this is a question mostly to you guys. By knowing the mass of the peptide, can we find out the sequence of the peptide? Actually, I heard many suggestions, like, for example, what you can do, you know the mass of this peptide, and you know which protease you used, and you can go by window, you can check all peptides which can be possibly generated from this protease, and you can find out the match. In reality, there's a case like this. Let's say we have 15 arginines and one lysine, let's say. And as a result, you can, whatever, it's of course a very artificial case, but you can take any combinations. And truth is that using this method, you will not able to distinguish one combination from each other. So one from another combination. We can for sure know C on N terminal. It's actually a question, how we can be, when we degraded protein with protease, how can we for sure know that this peptide is coming from C terminus and N terminus? Probably we can do this if we would somehow modify C terminus and N terminus of initial native protein. But it's actually not what's happening. It's usually this is additional step which people don't do. So in reality, this is a very hard question. So it's so there's no, let's say, software way to solve this. So, but there's a hard way to solve this. And maybe you know, like when physicians would like to know what's the there's a certain matter, and we would like to know what this consists of. We are usually kind of crashing this matter against something. So we're using this in the large Android collider, LGC, and many other places. Yeah, so that's exactly what's happening in this case. So we are doing some instrument inside instrument fragmentation. What I mean by that, we can somehow we can isolate certain species. We can isolate certain species of the peptides. Let's say we see from the first MS certain sets of the peptides. And now I'd like to say, okay, now I'd like to analyze this specific peptide with this specific mass to charge range. So we can trigger some activity in the instrument that this instrument will start collecting this isolation, like all peptides from this isolation window. After collecting all these ions, we can actually make fragmentation inside of the instrument. What does it mean? Let's say simple, there's many different ways to do fragmentation, but one of this, let's say the simplest one, we know that the peptide is positively charged. So as a result, we can really push with electrostatic field all these peptides in one direction. But we also can take some neutral gas, let's say argon, and we can push with some high pressure this argon in another opposite direction. As a result, whenever this species colliding, kinetic energy of argon would be enough to introduce one breakage into the peptide. So this is exactly what's happening. So we just, this fragmentation is so kind of fine-tuned in a way that statistically speaking, that should be just one breakage per peptide. And that's what's kind of we are getting at the end. We are getting all possible fragments. If you think about this, should be all possible suffixes, all possible prefixes of the initial peptide. Because, you know, breakage happening, then we have two pieces. This piece is called prefix, this called suffix. And that's we should somehow see these fragments. What we're doing after fragmentation, we're taking all these fragments which we are getting, and we are throwing these fragments into the mass spectrometry, mass analyzer. So you have to keep in mind that we are not getting just, let's say, one molecule of the peptide. We are getting milliards, let's say millions of the peptides. And this fragmentation is happening simultaneously. And this is already kind of, you may call this stochastical process where this fragmentation happening, etc. But it's a rather probabilistical process. It's not like there's peptides prone to break in certain locations. Let's say there's some amino acids after which breakage is never happening. But the truth is that in our lab, we constructed machine learning algorithm which actually solved this issue well. It actually can really predict where's the fragmentation happening and how tense this fragmentation is happening. Okay, one thing which I have to say, that's there's a different, when it comes to the question which peptide to select for the second fragmentation, there's a different answers. And the answers, like we can subdivide these answers, but even though now we can see that it's all the world's getting more complicated, but there's a three main elephants in the room. So one elephant called data-dependent acquisition, second way data-independent acquisition, and third one targeted. Let's call data-dependent acquisition. So data-dependent acquisition says that, okay, imagine we do see, like here we see simultaneously 4 peptides, but in reality, let's say we see 200 peptides. We remember we spent first 20 milliseconds for measuring first mass, but now we have 980 milliseconds to do what? To do this, exactly this, what I said, we're going to select peptide, we're going to fragment this peptide and measure this peptide. Of course, it's a very time-consuming thing, but we're going to do it not just once, not for one peptide, but let's say we're going to do it for, let's say, 20 peptides. We do have time, there's 980 milliseconds. Within this time range, we can measure, let's say, just 20 peptides. We can collect this, fragment, and measure. And repeating these three steps like 20 times. And that's exactly what's happening. But in the case of data-dependent acquisition, we are saying that we're going to select the high-intense peptides. So we're going to see this 200 peptides simultaneously. We see all the spectra. It's very complicated. But we're saying, okay, let's select top 20 by intensity, and let's gonna queue them for selection, fragmentation, and measurement. So as a result, like all the rest 980 milliseconds, we're spending on actually doing this, on analyzing this top 20 peptides. But it's actually what's usually looks like. So this is a heat map. Actually, on one axis we have master charge from 400 until 1600, but on another range we have time retention time, so time of leaving the column. And we see that the most of the peptides is kind of really initially we have very many many like small peptides, and at the end we have many large peptides. But as you see, like in the terms of master charge, but as we see, like instrument selecting them with small squares. Okay, cool. But data-dependent acquisition has one big disadvantage. One disadvantage is related to the spec. So imagine that we took HeLa cell line, digested cell line, we extracted peptides, and we prepared the sample, and we just split this sample on 2 epidorfs, and one epidorf we measured today, and the second epidorf we measured tomorrow. And the question is how results are going to be different between these 2 methods. The truth is that it's going to be actually is not, let's say, in some cases, we're going to have situation when some peptide in day 1 was selected for fragmentation and for the additional measurement, and the day 2 it was not measured. And let's say the reason is because today it was top 19 and tomorrow it was top 21, and that's why today was measured and tomorrow it would not be measured. And this is kind of a bit annoying, you may say, yeah, because as a result we are going to have this issue of the missing values. We see identification today, but we don't see identification tomorrow. There's a way to somehow tackle this issue, but we are not really the silver bullet. As a result, we do have many missing values, and it's a bit annoying. As a result, And this problem is well known in the field. As a result, there's two additional methods, and let's call the targeted method, which I would say very simple. Imagine a situation which is actually very typical in medical research. Let's say you had breast cancer samples, and it's well known in the field that you can approximately clusterize all this. You can clusterize all patients breast cancer on 4 groups. Let's say 4 groups, and they have some specific pattern of the expression. And let's say we measured all these, let's say 1,000 samples, 250 samples in each group. We measured with data-dependent acquisition. We had a lot of missing values, but we come up with some very smart statistical ways to actually find out some proteins which do helps to distinguish between groups. You can use support vector machine, you can use some more fancy method, but we come up with classification, and we come up with most important features of this classification. Cool. And now, after building this model of classification, you don't want to be, and you know that soon many more samples going to arrive. And these samples, doctors going to wait for the answer, which group this sample belongs to. And as a result, you don't need to measure all these 10,000 proteins anymore. What's the point of this? Because you know that based on this, let's say 15 proteins, you can nicely distinguish all four groups of the breast cancer. And this actually, what's happening now? You have, let's say, 15 proteins. As a result, let's say you have 60 peptides from all these proteins, because we're using shotgun proteomics. And you know the mass of these 60 peptides, you know approximately time range when we're leaving column, and you can really make targeted proteomics. You don't want to make this data-dependent acquisition like this, you know, like discovery proteomics, when you don't have some focus. You're kind of just searching for all possible proteins. You're really focusing on this specific 60 proteins, 60 peptides, and you have very reliable quantification of this 60 peptides, as a result, 15 proteins. As a result, you can really, we don't have any missing values. You don't need to come up with some dramatically complicated imputation method. You can really just use the target proteomics and have very reliable quantification of these peptides. Okay, cool. Targeted proteomics, it's not so kind of scalable. It's not so discovery in the sense of data-dependent acquisition. It cannot measure 10,000 proteins. It can measure way less, but very reliably. So data-independent acquisition, which tends against data-dependent acquisition, it's actually some sweet spot between these two poles, let's say. Data-independent acquisition, initially it can look a bit really strange. So, but what we are doing in data-independent acquisition, we are not like a sniper taking a certain peptides. Yeah, we are not like a targeted. We are just looking for a certain specific window all the time. What we are doing in data-independent acquisition, we're taking windows from, let's say, from 400 until 500, and all peptides which happen to be in this window going to be co-isolated, going to be co-fragmented, and it's going to be co-measured with a second MS, so with fragmentation measurement. So next, we're going to take 500 to 600, let's say 600 to 700, 700 to 800, etc., etc. So of course, usually we're not taking windows of 100 Thomson. Usually we are taking windows of smaller, but this doesn't change the picture and doesn't change the main problem, which you can probably already figure out. So remember, in data-dependent acquisition, we are getting spectrum with specific peptide. And as a result, we can, from this spectrum, suffixes and prefixes, we can somehow figure out which peptide made that. But the problem now that we're going to get huge amount of spectrums, very complicated spectrums from not just one peptide, but from several peptides simultaneously. And kind of to figure out which fragment peak belongs to which peptide is actually a very complicated task. And that's why DIA is, let's say, considered a bit more complicated. But now our group is successfully applied both methods. Initially, it was called, initial method was DDA, like that dependent acquisition, and its publication 2008. And that's actually why MaxCon is mostly famous for. Actually, the MaxCon is mostly famous for very reliable support for the mass spectrometry, proteomics related mass spectrometry data. But this year, actually, of course, it's happened not within 1 year. We work on this a lot of like these 2-3 years. We also enabled MaxDIA software, which allows to analyze DIA samples. It's exactly this samples which we are not taking the specific, like a sniper specific peptide. We are taking the whole ranges, co-fragmenting it, co-measuring, and there's this kind of very complicated data, but still we can deal with this data and we can deal it nicely. And of course, MaxDIA, what's a feature of this? That's we never ever skip any peptide for fragmentation. So all peptides gonna be fragmented, let's say, at least once. And that's actually very cool. It means that this data have capability of mining, data mining. You can really find out certain peptides, certain peaks, which you was not able to describe before, and like find out some quantitative values there. Okay, cool. That's how our picture looks like. So that's what we're doing in MaxQuant. We are just taking the spectrum. We understand we're understanding this spectrum coming from certain peptide. We know this peptide is coming from certain protein, and by this value, by intensity, which we are getting from first MS, how we can get intensity from first MS. Yeah, you can imagine actually that's spending this 20 milliseconds each second, we do see some peaks of the peptide. We do see peptide. And the truth is that if we are measuring very frequently this peptide, we see some 3D shape of each isotope. And we can really integrate these 3D shapes, and we can just measure basically volume of this 3D shape. And this volume is going to be exactly some kind of intensity measure for us. And that's actually what means quantification in the it's like peptide quantification feature, let's say quantification for now. What means identification in proteomics field? It's deciphering MS-MS. You remember I told you that we have all possible suffixes, all possible prefixes, but the problem that's happening here that's we're not uniformly distributed. Intensities of them are not uniform. As a result, kind of we really have to somehow deconvolute this spectrum, and there's 2 methods to do so. There's the de novo algorithms, the methods which don't know anything about samples which we're analyzing. We do know just masses of the amino acids. And what we're trying to do, we try to connect different peaks by distance between them, by a mass difference between them. And let's say if we have some mass between this peak and this peak, the mass difference is exactly mass of amino acids E, then we can connect them, and we can also connect with next one where we inserted mass like amino acids P, etc., etc., etc. So we can really try to connect all of them, and based just on information of amino acids masses, we can reconstruct the sequence of the peptide. And database-oriented algorithms, it's basically exactly the same idea which is happening in transcriptomics. We can let's say we're analyzing some unknown species, and we're analyzing transcriptomics of the species, and based on the intersections of the fragments using like different methods, we can really reconstruct the whole transcript without knowing which species we used. Or if you're, for example, using transcriptomics of the human samples, it doesn't sound actually very optimal way to do so. So what we can do, we can just align all these fragments, transcriptomic fragments, on the human genome and find out where's this fragments coming from. And this is called database-oriented algorithms. So in proteomics, basically, is absolutely the same. We're just taking the reference proteome file. We are digesting. We know that we use trypsin. Let's say we're digesting this the file on the peptides, and we are making theoretical spectrum, and we're putting all this spectrum into the database, and against which we're going to search later experimental spectrums. Okay, cool. So honestly, if you would like to know more, and especially if you were a bit confused with a DA topic, honestly, I totally recommend you to check out our YouTube channel, MaxCon YouTube channel, where we have MaxCon Summer School 2021 Summer School playlist. It's going to be some commercial probably. So, but also inside of this playlist, there's a MaxDIA part 1, MaxDIA part 2, and MaxDIA tutorial. And if you really you're interested in this type of analysis, I totally recommend you to go and check it out. So I will send you just here. Okay, cool. Okay, now what I'm going to discuss, the rest of my presentation, which is going to be not so long now, I'm going to discuss proteomic stories. So before we discuss like some general terms of proteomics, how it can be used, now we're going to discuss. So that, yeah, so proteomic stories. So from one perspective, yeah, maybe if you do have some questions about first part of the talk, please feel free to write it right now. I will read this chat, and I will answer them when we're gonna come. For now, I'm gonna describe a few application stories, but feel free to interrupt me. So, for example, multi-omics perspective on gene expression. So we know that's for the question how active certain genes, we can now answer on a level of not just one gene, but on a level of, let's say, thousands of genes. Now we can answer using at least like, let's say, 3 methods. So first method is transcriptomics. We're just measuring how many different RNAs do we have, fragments of this RNAs. Also, there's another method. Okay, another method is proteomics, of course, which we're discussing today. But another method is translatomics. So sorry for maybe confusion, but this translatomics is basically ribosome profiling. What ribosome profiling is doing is measuring how many different ribosomes do we have on a certain RNA. And this is basically, if you have a lot of ribosomes, you're getting a lot of fragments from this RNA. That means that you do have active translation this RNA. Some small amount, it means that it's not so active, etc., etc. So the question is, which omic correlates the best between each other? So each dot is going to be one gene, and let's say this dot, and for this dot we have expression on proteomics level, and we do have expression on translatomics level. Okay, let's make a macro view on this. So who is the best? Which two omics correlates the best? So we see this is transcriptomics and translatomics. Yeah, but there's also one factor which can be taken into account. We do have the same technological pathway. So we have the same technology which sequencing them is basically luminance sequencing. But okay, who is the second? And the second, as we may see, and the question, of course, also related to this, was which NGS omic is the best approximation for the proteomics. And we see that the next one is the best is translatomics. We see that translational information, like how many ribosomes we have on certain gene, is actually correlates the best with proteomics data. And the second one, which we see, is transcriptomics. So honestly, it's obvious that, yeah, because translation, how many ribosomes, it's very closer to the actually amount of proteins. It's like close information level to the protein level, and rather than transcriptomics. And it's actually in general a very beautiful thing. So just keep in mind all the time that when you're saying that using transcriptomics we approximate how many proteins do we have, it's not like it's approximately true with such a correlation coefficient. But the truth is that in case of NGS, we have toolkit which allows us to do this even better. And this is translatomics. This is ribosome profiling. Just keep in mind that. Okay. So if some questions, feel free to write. The next, called SILEC experiment. This is a very important topic, and it also was, let's say, was very important and very crucial for the first measurement of the huge measurement of proteomics, for the very first truly quantitative proteomics. So SILEC experiment was probably very popular 10-15 years ago. It's still popular for specific applications even now, but see that's label-free without any labels. Actually, method is winner. Anyway, so what's a SILEC experiment? Imagine that we do have cell line. Let's say it's a HeLa cell line, and at a certain period of time, we grow this on a petri dish, and we separated all cells which were growing on this petri dish on 2 petri dish. And one petri dish is going to grow with argin. And okay, I have to say this is a very important comment. That's HeLa cell line cannot generate arginine and lysine itself. It has to get these amino acids from background, from media. And this is, it's also true for most of the human cells, probably for all human cells, because we are getting these amino acids either from food, either from bacteria which are living in our stomach. Okay, so as I said, we have two petri dish, and why petri dish is growing with arginine, lysine 0. And what the 0 stands for? It means that we don't have any isotopes. We don't have any additional isotopes. All carbons are carbon 12, all oxygen is oxygen 16, etc. And all hydrogen, it's hydrogen, not deuterium. Okay, which actually, no, okay, let's say it's mostly carbon actually. Anyway, and in another petri dish, we add also arginine and lysine, but arginine has like 12 carbons, 10 carbons, I'm sorry, 10 carbons mass of 12. And all of them we exchange on carbon 13. And also about lysine, we did exactly the same. All carbons 12, which we had 8 of them, we exchange on the carbon 13. And now, whatever this cell is going to synthesize, it's going to integrate this heavily labeled arginine and lysine. So also important to say this, this heavy arginine and lysine, they don't influence anyhow on the physiology of the cell. So it doesn't damage anyhow the cell. And let's be also honest, this isotope small is stable. So we're not really quickly, we don't have short half-life. As a result, you have to be careful dealing with the samples, but anyway, it's okay. So, and as a result, you're incubating the cell lines with new media, let's say for 7 days. And after 7 days, you believe that, oh, let's say let's say 3 weeks. After 3 weeks, you do believe that all proteins which are supposed to be changed, which are supposed to be exchanged, all proteins in this sample, a red sample, we should supposed to have lysine, heavy lysine and arginine in all proteins. Okay, cool. And after this, let's say 3 days of incubation with such media, you are doing experiment. So let's say the red channel, you are adding some drug. And, with blue, like with cells which were growing on light amino acids, we were having happy life, and we are giving them some placebo. And as a result, after, let's say, treating cells after half an hour, we just destroying the cells. So of course, cells somehow reacted on that, changed expression of proteins. We now would like to know which proteins actually change the expression. So what we are doing for that, we are destroying cells and we extracting proteins. And what's really cool about this method, that we can actually, even after detaching cells from the surface of petri dish, we can mix the cells in one sample, in one Eppendorf. And you actually soon will understand why we actually can do that. But we are mixing them, and all processes of sample preparation, digestion, filtering, lysis, etc., we're doing in one all together. All peptides, all proteins are going to be generated all together in the same Eppendorf. Why it's cool? Because imagine like experiment can be not so robust. I don't know that you can add, you can treat protease like 10 minutes, and tomorrow you can treat protease sample by protease 11 minutes. Not seeing this difference, you can introduce some biases. But treating all them simultaneously, all these biases are actually uniform to one sample and the second sample. After that, you're taking this peptides and measuring with mass spectrometry. And honestly, what you're gonna see usually, you're gonna see a picture like that. So this is time range, this is mass to charge. And you're gonna see that you constantly have a situation when you have two peptides going simultaneously out of column. And this peptide and this peptide. So this is light peptide with light arginine and lysine, and this with heavy arginine and lysine. As you see, column chromatography cannot distinguish one from another. Who can distinguish is just mass analyzer. And we do all the time see a situation like that. And what's really cool now, we can really compare intensities of light channel and heavy channel. And in a situation like that, we see that some peptide X haven't changed anyhow concentration, but peptide Y changed concentration. And that basically means that protein Y also changed concentration. And you see that in the case of the adding drug, this peptide as well, protein, increase expression 3 times. And by that, we can do some differential expression analysis. Okay, one important thing which I forgot to tell you, that's we're using arginine and lysine not by chance. It's actually, we're using them because we're using trypsin. So as you know, trypsin, it's protease which is digesting just after arginine and lysine. And as a result, we can guarantee that every peptide in our sample going to have arginine and lysine at the end. That means by default that each peptide going to be in pair with heavy peptide and light peptide. So it's all good all the time. All peptides, all features on the first MS spectrum going to have two features, heavy and light. Actually, maybe I forgot to tell you one important thing, actually. So the mass spectrometry, shotgun mass spectrometry, which we're discussing here, called another name for that is MS-MS, and another name is tandem mass spectrometry. Tandem, it just means that after first MS, we're doing second MS. And as you remember, first MS, when we're measuring fragments, and the second MS, when we're measuring, first MS, it's when we're measuring peptides, and the second MS, when we are measuring fragments of specific peptides or set of peptides in the case of DIA. So first MS, peptides, second MS, fragments. That's why it's called tandem mass spectrometry. That's why it's called MS-MS, MS-MS proteomics. Okay, cool. Okay, how it can be used? How this Silek experiment can be used? So honestly, initially, as you can imagine, it was used as a very sensitive differential expression method. So you can just treat one cells and keep control of other cells and can measure and see which proteins actually change the concentration. Okay, but there's some specific methods, a specific application where the Silek method is incredibly powerful technique. Let's say one of them is actually a half-life measurement. So imagine that at a certain moment zero, cells were growing, no problem whatsoever, and moment zero, it's a moment, time point zero, when we changed media with light version of arginine and lysine on heavy version of lysine and arginine. As a result, all proteins gonna accumulate, all cells gonna synthesize new proteins, and these new proteins gonna incorporate heavy arginine, heavy lysine. But the question is how quickly we're gonna do this. And this is actually what I mean by half-life. So, and let's check for this example. So we're measuring not just once, we're measuring after 1.5 hours, we're measuring after 4.5 hours, and we're making third measurement after 13.5 hours. So, and let's look at a certain peptide. You remember I told you that in case of Silek, each peptide have to be paired. And the result, we see that. We see some peptide from certain protein RRM2, we see some light peptide and heavy channel. And we see that after 1.5 hours, light is still the most abundant. But after 4.5 hours, we see that heavy peptide is getting larger. So this exactly means that something around 4 hours, it was the moment when the light channel was exactly the same intensity as the heavy channel. 4 hours to half rotate the proteins, half exchange half of the proteins in your cell. But after 13 hours, we see that we do have almost nothing from light channel, almost nothing. We have a lot from the heavy, but almost nothing from light. So almost all proteins were exchanged after 13 hours. So, okay, half-life of this, half synthesis of this protein is basically 4 hours. But we can look at another peptide, let's say this peptide coming from histone, and we see that this equilibrium is happening approximately in 15 hours. Yeah, we can approximate it, and we see that this is the balance half-life ratio 0.5, half-life ratio 1, going to happen just like after 15 hours. It just means that this protein really very slowly exchanged. You see, this is actually a very important thing. So we kind of looking at the expression as a static picture, but of course it's not static. Some proteins are heavily synthesized and destroyed. This kind of space is very important for it can be very important for certain regulation. It's actually, we can also measure this for RNAs. There's another technique to measure also half-life of RNAs. It's not actually very complicated, but measuring two things simultaneously, it's what's actually was the distinguished feature of this paper. And actually totally recommend you to read this paper. It's by many senses actually was a breakthrough in the field. There's many interesting questions which you may ask, like is protein with very short half-life and also very short RNA half-life? Is it true or not? So yeah, all these questions actually incredibly interesting, and but I'm really referring to this paper for you to know more. Actually, honestly, let me do one thing, and also maybe I will share just right now the link. You can later use it, and I will post it once more. Okay, cool. Protein and RNA half-life. Incredibly cool method. I saw many articles related to this method. It's incredibly powerful. Okay, again, if you have some questions, feel free to ask. Okay. Okay, what's more? So you also you can measure multi-omics of Slavic microbiome. It's not a related topic to the SILAC, but it's rather related to the, let's say, microbiome topic. So what you can do, you can just, in this example, in this study which I was a part of, we just measured different saliva samples, so from the mouth. And we found out that 50% of the peptides which we see were not coming from the host, not from the human. But 50% of peptides which you see are coming from some bacteria. It's no surprise, of course. But the question which we were raising, actually, can we quantitatively answer the question, how many different bacteria do we have? So how many, do we have some, I mean, maybe we do have some bacteria in a larger proportion than another one. And of course, what we would also like to do, we would like to compare these proportions to the whole genome sequencing project of the human microbiome, human microbiome project, which was doing this not with mass spectrometry, not with proteomics, but rather with sequencing of DNA samples. And what we did there, we somehow very complicatedly normalized samples, and we did just PCA plot on the spectrum of the bacteria which we see on mass spectrometry and on this whole genome sequencing of human microbiome project. And we see that we do this human microbiome project has different body sites, which includes mucous, mouth, gingiva, nose, trees, vagina, and stool. And we see that our samples from mass spectrometry are actually located where it's supposed to be located. So we're really located in the mouth, in the mouth and mucous. It just really supports the fact that the mass spectrometry do detect peptides, and these peptides, even though it's very hard to subscribe to special stem of bacteria, it's actually a very complicated task. But even though we can go to the family level and measure, like checking how many different peptides we found for each family, using this information together with whole genome sequencing, we can actually see that the data are well correlated. So our proteomics data are coming from body size which we're supposed to come. This was just a proof of concept, but I think it's actually beautiful and like showcase that proteomics can do that. Okay, cool. Another story. I'm going to be quick now. So cancer T cells and two smoking immunopeptides. So this is actually a poster which we made for like internal meeting in Max Planck Institute, but this suggestion was actually cancelled, and nobody, yeah, so our administration didn't make a poster out of this. Okay, cool. So we know self versus non-self, how our adaptive immune system works. So our cell have expressing on the surface MHC1 complex. And this MHC1 complex representing some random usually 9 amino acid peptide from inside of the cell to the surface of the cell. And this 9 amino acids peptide can be detected by T cell, and this T cell, when recognizing non-self peptide, start destroying this peptide, which representing this start destroying this cell, which representing this peptide. And why it's doing so? Because I mentioned that we have some viruses which are invaded cell, and as a result, the cell not anymore friendly cell of our body. This is now a factory of which producing viruses. But our body still can recognize that something going wrong, because this MHC complex can really represent one random peptide from this viral protein. And this immunopeptide is going to be recognized by T cells, and T cells kill this cell way in advance. But what's true is that the system not just working against viral peptides, it's also working against cancer. And because cancer really accumulates huge amount of non-synonymous mutations, and these non-synonymous mutations can be represented, can be expressed, and this expressed non-synonymous mutations can be represented on the surface. And if this non-synonymous mutation is very specific for this cancer, not for the rest of the cells, then the immune system has some probability to detect it and kill it. So the thing is that in proteomics, what we can do, we can really just wash out all these peptides from the surface and detect them, and detect some peptides which do have non-synonymous mutations. And why it's cool? Because these peptides, if we find some such peptides on the surface, we can actually really synthesize them chemically, and we can train the immune system against this peptide. Or we can really just infuse this peptide into the blood and train the immune system inside of our body. There's different possibilities. For now, we just take the cancer samples. We, of course, had the PBMC, so blood samples. We were measuring exome sequencing from them, as well as from the tumor, and we were finding mutations which had this non-synonymous nature and which had also somatic nature, or so basically means that these mutations are specific for the tumor. And we were including these mutations in our database search. And from another perspective, we were taking proteomics and we were measuring peptides which were presenting on the surface. And taking these peptides, using the spectrum, we try to find some peptides with mutation, and we do find something. It's actually, we usually find, like, let's say 1, 2, 3 peptides per patient, and it's actually already a lot. The proof is that we can just take this peptide and find out using an immunogenicity assessment that there's a reaction of immune system on this peptide. The another question, why immune system not killing the cancer? There can be some immune the checkpoint blockage drugs can be useful here, but this is another topic. So why melanoma? As you know, melanoma is the most, like the cancer which accumulates huge amount of mutations. As a result, we just increasing probability that some of them are going to be not synonymous. This was a proof of concept. So the cool thing about this method, that's usually this experiment's done without any proteomics. We're just done with NGS, let's say exome sequencing of the tumor and exome sequencing of the blood samples. You're comparing them, finding some non-synonymous mutation, and next, what you're doing usually, you are taking, you're sliding through this non-synonymous mutation with like 9 amino acids, and you're taking these 9 peptides with mutation, and you have some usually machine learning tool to predict how well it's, how strong affinity this peptide to the certain MHC complex on the surface. And as a result, if like there's a strong affinity, this would mean that there's a really higher chance this less peptide going to be present on the surface of the cancer cells. And but the problem here that's in this case, this process of the representation is not actually so simple. So, and to have some actually direct evidence that this peptide present on the surface, it's actually a great advantage. Actually, situation right now is converted a bit in a way that this algorithms of machine learning prediction to be present or not present is actually improved with proteomics data, which we're having right now. It's actually another topic, but this is actually, I think, the way to go. But yeah, so let's skip that. This actually it's really important to highlight. So the thing is that our body, each cell of our body has 6 alleles of MHC complex. As a result, you have 6 different sets of the MHC peptides which are present on the surface. So it's actually a very complicated data set. You're getting some peptides, but you don't know from which MHC is coming from. So it's really not good for the training, any machine learning. So what people did, and actually these 2 papers for Stephen Carr group, I totally recommend you to read that. So we used a single allele. What we did there, we took some cell line, moved all MHC complexes, all 6 MHC complexes from that, and we inserted, let's say, one certain specific MHC complex. As a result, we measured all peptides from this MHC complex. And next, we took the cell line and inserted the second MHC complex. We were doing this already for now for hundreds of cell lines, and by that, we're able to measure like really this information, which is super perfect for the machine learning training. You have MHC complex, specific MHC complex, and you see some specific and you see some patterns, and you can make some prediction. Okay, cool. This was the second poster actually, which was also not accepted, but this was one flew over immunopeptidomics, and I hope you like it. Okay, cool. Okay, maybe I will shift that. Okay, let me know. Let's say 12 minutes. I probably will describe this topic. It's actually a very interesting experiment. It's called the thermal shift experiment. So what this experiment is doing? So let me tell you what it's doing at the end. At the end, okay, what's the question? The question is, I have a cell line. I have a huge amount of different proteins in this cell line. So my question is, and I have some drug. I'm a pharmaceutical company. I'd like to know with whom, with which kinases, let's say, with which proteins this drug is interacting. And why it's important? Because imagine I was doing huge bioinformatics pipeline with docking certain drug to the certain protein, and I really somehow sure there's this very specific interaction. But the way to actually really check that is will be some macro method to take the drug and see which proteins do interact with this drug. And it was several iterations of mass spectrometry way of thinking about this problem. And actually one of the direction which I think is super successful now, it's thermal shift experiment. So what this experiment is doing? So imagine that's we have two epidorfs, and the one epidorf with lysate of cells with placebo drug, and another tube we have actually a lysate of cells with our drug of interest. So what we're doing next, we are incubating these two tubes in temperature of 37. So let's say we don't have, we split these tubes on the, let's say, 10 different tubes. Okay, with first set of the tubes, we just incubated with 37 degree, and we measured all proteins which stays in media. What I mean by that, some proteins will collapse and fall on the bottom of the epidorf, and some of them not. And exactly the proteins which didn't fall in the bottom of the epidorf, we're going to measure. And next, we're taking the second set of the tubes we're incubating with 41 degree, and whatever haven't collapsed, we're measuring. Let's go to the extreme. The last temperature, 67, is really already high temperature, and almost all proteins are collapsing no matter what. And the result, let's check. Yeah, I'm sorry, that's like an additional experiment called TMT, but it's not actually very important. What is really would be important to see, it's something like that. So imagine that when we were treating cells with some vehicle, this basically means placebo, some unrelevant drug. We see that this protein was in highest concentration in temperature 37, but you see it's with increasing temperature, this protein starts collapsing to the bottom of the epidorf. And you see it's on temperature 56, 60, almost 100% of protein collapsed on the surface. Okay, but the cool thing about this, that's when we add certain drug, we see that this protein getting more thermostable. So we see that this 100% dropping down is happening already, let's say, after not like 60 degree, but after 65 degree or something like that. In order to be quantitative, we can actually measure temperature when half of the protein collapsed to the bottom. This is going to be our definition of melting point. And what we can do now, we can really compare how melting points change thanks to adding this drug. And what's really cool about that we can do this for 10,000 of proteins simultaneously. We really can do this measurement. We can measure how thermostability changed of the 10,000 of proteins. And the truth is, if drug can attaching to certain proteins, then this protein is usually getting more thermostable. And the reason is because this drug is bringing some connections, some bonds. It can be hydrophobic, whatever bonds. But the thing is that it's making protein more robust against like destruction, against unwrapping, and against the melting, and etc. So as a result, melting point of proteins with drug will be increased, and by measuring how strongly this temperature shift happened, thermostability happened, we can actually really understand with whom this protein interacts strongly or not less strongly, etc. And this is actually extremely powerful. Imagine we can do this for 10,000 proteins simultaneously. And we can, for example, there's some drugs, stausporin, and we see that this drug interacts with huge amount of different kinases in the cell. And you can imagine the specificity of this drug is not really high. As a result, if this drug was developed against certain kinase, it really interacts with many kinases. As a result, you're going to have a lot of side effects. And this is really not a good drug. But if you look at another drug, let's say harmine, you see that the kinase with whom this drug is interacting is DIRC1A. And this really shows you that this is the major interactor with this drug. And we did this by no bias, prior bias. We just measured just 10,000 proteins and just measured the smelting curve of these proteins. Extremely powerful method, and it's used a lot. Okay, proteomics, one of the gold mine of the proteomics is also protein-protein interactions. And one of the simple things you can do, you can just attach certain tag to the protein X, the protein which you would like to know with whom this protein is interacting. You can attach, let's say, tag GFP. Of course, it's a huge tag, but it's very easy to get because, many people did microscopic measurements of many proteins. Anyway, so we have some specific antibody against this tag. Of course, we may have a very specific antibody against protein X, but it's not a very scalable solution. So anyway, what we are doing, we are incubating this fusion of protein X and GFP, incubating inside of lysate or inside of cells. And next, we just pulling this protein using this tag and checking out with whom this protein is interacting, with whom protein X is interacting. Of course, you can find out that you can do it not just for one protein X, you can do it for protein Y, and you find out that background stays the same, like non-informative interaction, but informative interaction is gonna vary from protein to protein. You can do replicates, etc., and you can do it not just for 1-2 proteins, but you can actually do it for 1,000 proteins. And actually, current updates is people doing already several thousands of proteins. It's actually extremely powerful because you actually can really see something like that. You can see that you can make a network analysis. You can see that's one protein interacting with another protein. If you're pulling protein X, you see protein Y. If you're pulling protein Y, you see protein X. And you can do it with massive scale, and you can make this protein-protein interaction networks. You can see this protein-protein interaction networks depends from some environment. So it's incredibly cool field, and it's actually actively developing. It's also actively developing, for example, in the case of virus. You can see with whom coronavirus is, with which proteins certain specific coronaviral protein is interacting. Like whom this protein is targeting being the host proteome. It's actually done by many groups now, and it's actually cool research. Okay, now I'm going to be again very quick. So one thing which I would like to also highlight, one method, is called protein localization in the cell. Currently, that's protein localization we can measure using fluorescence microscopy, but the problem with this method, it's not scalable. So we can measure if you, let's say you have some process, I would like to know which proteins do change localization, then you have to measure whole proteome. You have to label whole proteome and one by one see which proteins do change localization. That's a very not scalable solution. But what you can do, you can apply some proteomics method. What you can do here, imagine we are taking sucrose gradient and we are destroying cells. We are slightly destroying cells, means that we are destroying outer membrane but not internal membrane, not organoids, and we're putting this soup on the top of this gradient. Next, we are rotating with one speed, and something super heavy and something compact falling down. So usually it's nucleus. And next, you're increasing speed, and okay, sorry. And whatever didn't fall down, you're transferring to the new tube. And whatever fall down, you are gonna send for the measurement, mass spectrometry measurement. And next, you're increasing speed up to 3K and repeating this cycle, and next 5.4K, 12.2K, etc., etc. So at the end, something really light and not condensed will start falling down. Okay, what's cool about this method? So the cool that's, if you think about that, you're measuring organoid proteomes, you will have concentration of protein over the 6 fractions. So imagine that certain protein is really heavily localized in mitochondria. Then it's going to be in first tube is going to be nothing, in second tube is going to be a lot, in third tube is going to be way less, and fourth tube is way less, and this is almost disappearing from the rest of the tubes. And if protein, for example, had let's say some lysosome, then concentration going to be distributed differently. So the highest concentration going to be in fourth tube, but not